Okay. So today we're going to start this new topic on the endocrine system. And basically what we're going to be discussing here is the idea of intercellular communication, especially intercellular chemical communication. And again, what we're talking about here is how one cell communicates to another through chemicals. And we're going to be talking about the release of these, and these chemicals are going to be hormones. We're going to be talking about the release of the hormones. We're going to be talking about the binding of the hormones to the receptor, and how the receptor hormone interaction, that complex, can then affect changes in the cell. That's largely where we're headed in the next couple of days. We're going to start out here with some simple ideas of communication in terms of autocrine, paracrine, and endocrine. And in autocrine, the very same cell that's releasing the hormone is responding to it. This is simply saying that the hormone that's, This is simply saying the cell that is secreting the hormone is also the one that's responding to it. In a paracrine response, it's the nearby cells that are responding to the released hormone. And in the endocrine response, which is what we normally think of in this whole system, is that this is really much longer distance communication. Where this is the same cell, this is the nearby cell, the endocrine response is hormones traveling through the blood to cells that are far, far away, and those cells responding by binding to those hormones. Well, what this means is there has to be a receptor in all of these cells, and there has to be a molecule that's released. The molecules released, we're talking about the hormone. The receptor is the molecule to which the hormone responds, and that will then elicit a response in the responding cell, and we'll walk our way through that. There are a fairly small number of molecules that we can use as chemical signals. The types of hormones, the first one I'm going to tell you about is a peptide or protein hormone. What I want you to hold on to from the idea of peptide or protein hormones is they're large, they're charged, and they're hydrophilic. So we're going to see that one of the characteristics of all the peptide or protein hormones is that they have to communicate to the inside of the cell through a transmembrane protein. It's going to be a transmembrane protein. We're going to talk primarily about G-protein coupled receptors, but these are transmembrane proteins that bind the hormone on the outside of the cell and signal to the inside of the cell, causing a cascade of events. The other types of hormones we'll have in our bodies are modified amino acids. And there are only two amino acids that we're going to modify into hormones. The first one is going to be tryptophan. Tryptophan gets modified into serotonin. This is important not only in the brain, it's important in a number of places. We're not going to talk much about it. We're just going to say that serotonin is a hormone and that it's modified from tryptophan. The other kind of amino acid that we can modify to make hormones is tyrosine, and this actually is much more common than tryptophan. Tyrosine is modified into the so-called catecholamines, dopamine, epinephrine, and norepinephrine. And we're going to talk about these in the next segment when we talk about the neurotransmitters. These are all neurotransmitters, but they also can function as hormones. They're all modified from tyrosine. All of these catecholamines are hydrophilic. So the catecholamines are hydrophilic, and that's in contrast to the thyroid hormone. The thyroid hormone is made in the thyroid gland. Its fundamental structure is derived from tyrosine. But this modified tyrosine, once it becomes thyroid hormone, now becomes a hydrophobic hormone. And one of the ideas we'll run into here is we're going to contrast the ways that hydrophilic hormones and hydrophobic hormones work. It's a bit of a generalization. It's not always true. But by and large, when we talk about a hydrophobic hormone, such as thyroid hormone, or the steroid hormones we're going to talk about in a moment, those all function through cytoplasmic receptors. And logically, the way this functions is the thyroid hormone and the steroid hormones, because they're so hydrophobic, are easily dissolved in the phospholipid bilayer. So they easily pass through the phospholipid bilayer into the cytoplasm of the cell, where they can then bind to a receptor. That's in contrast to, say, the catecholamines or the peptides, all of which are extremely hydrophilic and therefore can't pass through the phospholipid bilayer. To activate responses in the cell, they have to work through a transmembrane protein receptor. The other category of hormones we're going to talk about are the so-called modified cholesterol hormones. Typically, these modified cholesterol hormones we subsume under the heading of steroid hormones. So all of these steroid hormones, whether they're talking about testosterone, estradiol, progesterone, calciferol, which we talked about yesterday in terms of vitamin D, cortisol, or corticosterone. Should be an R. The hallmark of all of these hormones is they're hydrophobic. They are generated from a cholesterol molecule. The cholesterol itself is hydrophobic. We put all manner of side chains on them and make them into different kinds of hormones, but fundamentally they're hydrophobic. Because they're hydrophobic, they easily pass through the phospholipid bilayers of cells and then will bind to receptors in the cytoplasm. So we'll spend a lot of time comparing and contrasting these cytoplasmic receptors with the transmembrane protein receptors. There's one more in here. I'm going to mention it. I'm going to put it down at the bottom because I forgot to put it in my list, and that's modified fatty acids. These modified fatty acids are mostly prostaglandins. You'll run into prostaglandins late in the course when Dr. Bauma comes in and talks about parturition. Parturition and the activation of the myometrium depends to a large extent on these prostaglandins. If you get into the nervous system and some of the second messengers, you'll talk a little bit about prostaglandins there as well, particularly arachidonic acid. We're not going to get very far into these. I'll just tell you they're modified fatty acids that generate prostaglandins, and we'll leave it there. But regardless of which of these hormone types we're talking about, regardless of what receptor types we're talking about, whether they're a transmembrane protein or a cytoplasmic protein, what's characteristic for all of them is there has to form a receptor hormone complex. And the rules of the formation of a receptor hormone complex are just basic chemistry. They're the law of mass action. These are not covalent changes. These are charge-charge interactions. And so they're really akin to the binding of an enzyme to a ligand, or enzyme to a substrate. But we can talk about it in terms of concentrations, where we have the concentration of hormone plus the concentration of receptor. These brackets are telling you its concentration. That gives rise to the hormone receptor complex, but this is not a permanent arrangement. The hormone receptor complex can become dissociated, can come back apart into its hormone and receptor. When we think about the generation of these hormone receptor complexes, there are a few ideas I want to introduce here. And if you take another chemistry course or another biochemistry course, you'll get much farther into the weeds on this. But I just want to introduce a couple of ideas here that will be important to understanding how this hormone receptor complex functions. First, when we think about the endocrine system, we have to recognize that we have a fairly small number of sending or secreting cells, no more than maybe a few thousand in many instances. Those few thousand cells will produce, maybe a few million molecules of hormone, which sounds like a lot, but when you dilute that few million molecules of hormone throughout the body system, or among trillions of cells, those concentrations fall rapidly. So what we find with the hormones is that there needs to be a high affinity. We need to have stickiness, for the most part, for the hormones to the receptor. So that when we have these exquisitely low concentrations of hormone, which can often be 10 to the minus 9th to 10 to the minus 12th molar. That's nanomolar to picomolar. These very low concentrations still need to bind to a receptor. That means these receptors need to be sticky. So that means they have to have high affinity. But having high affinity for the receptor isn't adequate. We also need to have specificity. These hormones need to fit into the binding pocket of the receptor in just the right way. We need to have a three-dimensional fit between the hormone itself and the receptor. So we have these ideas here of affinity and specificity. Along with the idea of affinity, we can talk about the dissociation constant. Dissociation constant is something out of physical chemistry. It's denoted as Kd. We're going to simply define Kd as the hormone concentration at which half of the receptors are occupied. And we'll work a little bit with that. And if we say the Kd for a hormone is 10 to the minus 9th molar, That means when we have 10 to the minus ninth molar hormone, and if we have adequate numbers of receptors, half of those receptors will be occupied, and we'll walk our way through what that means. But let's start here with some simple ideas. And when we think about the endocrine system, what we're thinking about is chemical communication between cells. Again, if you imagine all of this story evolutionarily, once multicellular organisms begin to evolve, we have three-dimensional organisms. They have thousands or hundreds of thousands or millions or trillions of cells. There needs to be a way of communicating between these cells. And so we use the endocrine system to form an intercellular chemical communicating a chemical communication system. Now, that's a fairly arcane thing to say. Endocrine system is an intercellular chemical communicating system. But fundamentally, when we talk about this, we have a sending cell, and we have a receiving cell. The sending cell is going to be secreting hormone. The receiving cell is going to be responding to hormone by binding hormone to a receptor. There's a subtlety here that I really want to emphasize, and that is if a cell has no receptor, the hormone can't signal it. The only way the hormone can signal to a cell is through a receptor. It doesn't matter in this case whether the receptor is in the cytoplasm or across the membrane. The only way this signaling cell, the sending cell, can send a piece of information to a receiving cell is if the hormone being sent by the cell can bind to a receptor on the receiving cell. So a simple idea, if you have a sending unit and a receiving unit, the receiving unit must have a receptor that's specific for the hormone that's been sent by the sending unit. So what we're going to do over here then is we're going to think a little bit about the idea of types of intracellular. I should point out this means between. Communication. We can think of this intracellular communication in three ways, but it will always obey the rule that you have a sending cell and a receiving cell, and to be a proper receiving cell, you have to have the receptor that will bind to the hormone. And the first one I'm going to put up here, I'll just put this in blue. This box is meant to be a cell. And this cell can secrete hormone. So through exocytosis, the hormone can be released. So this is secreted hormone. Now, if this cell has receptors on it, and I'll show this is transmembrane protein receptors. If it has receptors specific for the secreted hormone, the cell can respond. Well, this is a simple case. What we're saying here is this cell is the sending cell. It's the sending or signaling cell. But the very same cell that's sending the signal can also respond or receive the signal. And so we can imagine these hormones, these molecules that have been released binding to those receptors. And this has a particular response. It's called autocrine. Autocrine meaning self. So this is a self-responder. And there are any number of cells that do this. They release a hormone and they have receptors on them, and this amplifies the response of the cell to that particular hormone. There's another one I'll show you here on the right-hand panel. I'll show some green cells here. And this response that I'm going to label on this panel is called the paracrine. Para meaning around or about. And in a paracrine response, what we'll find is there will be a signaling cell. I'll mark this as the signaling or secreting cell. You call it a sending cell. You call it a secreting cell. It's conceptually all the same. It's releasing a hormone. So it's releasing a hormone here by exocytosis. But now this signaling cell itself, unlike the autocrine response, this signaling cell does not have receptors for the hormone. The receptors of the hormone are on another one of the cells. And so here's another cell nearby. It's not the very same cell. It's a nearby cell. And this now is the receptor for hormone released by a nearby. It needn't be neighboring. It simply needs to be the neighborhood. Nearby cell. So here's the nearby cell. It's secreting hormone via exocytosis. The hormone is out in the extracellular space, and it diffuses around, and it binds to these receptors. It can now affect a response in this cell. This is a paracrine response, para meaning around. We'll run into this particular response when we get to the epithelial cells of the stomach lining. And we'll see that the acid-secreting cells, the parietal cells in the lining of the stomach will respond to a hormone called histamine in this paracrine fashion, just to give you an example. On the left-hand panel, let me show you the response, which we normally think of in terms of an endocrine response. A true, what I'll label up here as a true endocrine response. Now, how I want to differentiate this, what I'm calling a true endocrine response from the other two, is that the hormone is secreted into the blood. And once in the blood, it can be carried throughout the body, the system. So we can think about this a little bit, and I want to imagine here a blood vessel, and I'm going to show the blood vessel in a little more detail and say that these oval structures are meant to be the endothelial cells. Lining a capillary. We'll see later that the capillary is a kind of blood vessel. We frequently call it an exchange vessel. They're exquisitely thin. They're composed of a single layer of these endothelial cells. Well, nearby, we can put in red, another cell over here. This is going to be the secreting or sending cell. It secretes a signal. The signal is a hormone. But now the hormone doesn't remain in the same environment as we showed for both the paracrine and the autocrine response, but rather the hormone gets into the blood. And it's swept away in the blood so that a cell, a blue cell down here, far, far away, this is going to be the responding cell. For this cell to respond to this hormone, as we've said, it needs to have some kind of receptor, so that that hormone can bind to these receptors and cause a cellular response. Now, I think you can see if this is a cell, perhaps in the anterior pituitary that we'll talk about on Monday, and these hormones are being released and they're going out to their target gland, this small number, maybe a few million molecules of these hormones, now have to find their way to receptors on a specific set of cells, because only those cells that have the receptors to which the hormone can bind will show an effect. This is going to be a subset of cells in the body, and what's more is they will contain maybe a few thousand receptor molecules. So what this means then is the hormone concentrations typically act at low concentrations. The concentrations where you'll typically see these things function is about 10 to the minus 9 to 10 to the minus 12th molar. There are a few that even can work in lower concentrations, down almost to 10 to the minus 15th molar. But for the most part, they're in this 10 to the minus 9th, 10 to the minus 12th molar range. You'll see some effects all the way up into micromolar, which is 10 to the minus 6th. And we'll talk a little bit about that perhaps by the end of today. But the idea here is you have to have a receptor that's specific for the hormone, has to bind the hormone, and then affect a response. So any questions up to there?
How does the hormone get past the endothelial cells?
Great question. How does the hormone get past the endothelial cells? In some cases, there will actually be gaps between those endothelial cells. So that we like to think in many cases these endothelial cells are all zippered together with tight junctions, but not always. Many times there are small gaps here between adjacent endothelial cells. We'll see later on, some of these gaps could be quite large, but these hormones have molecular weights well less than 10,000 for the most part. And so they can easily pass through narrow gaps and get into the blood vascular system. It doesn't take much of a space between adjacent endothelial cells to provide a large enough gap for the hormones to get through. Anything else? No. Well, what I want to do here is I want to think about what the hormone can do to the cell. Now, I'm going to tell this story from the point of view of a transmembrane protein and a hormone binding to that transmembrane protein. But we could pretty much tell the same story if they were cytoplasmic hormones. But the transmembrane protein hormones are more common. So I'm going to tell the story from the point of view of a transmembrane protein hormone a transmembrane protein receptor binding to the hormone, and explore what kinds of changes that this hormone receptor interaction can cause in a particular cell. So we'll simplify this a lot. I'm going to make the cell a box. I'm going to make the transmembrane protein a simple slotted structure so that we can imagine it binding the hormone, and then just think about what can happen downstream to the hormone receptor complex formation. So this blue line is meant to represent the plasma membrane. And across the plasma membrane, of course, there will be a transmembrane protein receptor. I'm going to show you this is just sort of a V-shaped structure. This is meant to be the hormone receptor. There needs to be a good fit between the hormone and its receptor. So I'll just sort of show this as a kind of triangle, meaning the hormone. So this is the hormone. And taken together, we have generated a hormone receptor complex. What will happen after the hormone binds to the receptors is there'll be a whole series of downstream effects. After the binding of the hormone to the receptor, we can get a number of things happen. And I'm going to spell them out on the right-hand side and then try to illuminate them or illustrate them in the drawing. The first thing that can happen is that the hormone receptor complex can alter enzyme activity. It can increase enzyme activity or it can decrease enzyme activity. But the binding of the hormone to the receptor can, inside the cell here, affect enzyme activity. In addition to affecting the enzyme activity, it can modulate ion channels. So this is meant to be an ion channel. So in addition to altering enzyme activity, the second thing it can do is that the hormone receptor complex can alter, you can read modify, ion channel activity. It can change whether the channel opens, it changes how hard it is to open the channel, it can change whether or not it closes. There are a number of modifications it can make, but it will alter or modify ion channel activity. That will then change or regulate the movement of ions into and out of the cell. The third thing that it can do, so we can imagine it here modifying ion channel activity, the third thing it can do, let me put this in green, is that it can modify or affect endocytosis and exocytosis. It can modify the rate, for example, at which we fuse vesicles to the plasma membrane and incorporate the proteins into the plasma membrane. So if we imagine here in orange, we have some transmembrane proteins in the vesicle. These are now going to be incorporated into the plasma membrane. So now we can regulate the number of these transmembrane proteins that are in the plasma membrane by regulating the probability of endocytosis or exocytosis. We're going to see this is a particularly important phenomenon in the kidney, that there's a hormone called vasopressin, which is going to regulate the incorporation of aquaporins into a particular set of cells in the kidney. And that's all done by regulating exocytosis and endocytosis. So it's an important enzyme, it's a very important function of hormones. The fourth thing that a hormone can do is it can regulate or modify gene transcription. And translation. This is a fancy way of saying that the hormone receptor complex has the possibility, once it's formed, of sending signals to the cell nucleus. This is the nucleus. So the receptor hormone complex formation can send signals down to the nucleus to turn on or turn off genes. So we can now regulate protein synthesis in these cells. We can regulate which proteins we're going to make or which proteins we're going to turn off. And that can be done through regulation of gene transcription and translation. So again, when we think about this intercellular communication, there are fundamentally only 4 ways the hormone receptor complex is going to affect the physiology of these cells. It's going to affect it through enzyme activity, channel activity, exo and endocytosis, and gene transcription translation. Those are the 4 things that the forming of a receptor hormone complex can do to the cell. Any questions to there? Okay. Well, if not, let's go to the right-hand panel. And we're now going to drill down and talk a little bit more about the kinds of molecules that can function as hormones. The kinds of molecules that can be secreted by a signaling or sending cell and then communicate to another cell. And there are a relatively small number of these kinds of signaling molecules. And we can take this panel and fundamentally label it as the hormones. We can think of them as these are the signaling molecules. Now recognize, for these to signal properly, they must work through a receptor, but they are still the signaling molecules. And they come in a fairly small number of varieties. The first one I'm going to tell you about is the so-called protein/peptide hormones. We distinguish between a protein and a peptide simply on size. When they get down below, I think it's around 50 or so amino acids, we call it a peptide. When it's bigger than that, we call it a protein. It's a pretty vague terminology, but peptides and proteins are very similar because they are constructed from sequences of amino acids. No surprise, because they're constructed of sequences of amino acids, sometimes a few hundred of them, sometimes as few as 9 or 10. These tend to be large, and they tend to be hydrophilic, meaning they're secreted from cells, easily go into the blood, go into solution, extracellular environment. They're hydrophilic. These protein or peptide hormones, because they're large and hydrophilic, will signal predominantly through transmembrane protein receptors. In here, when you think about a hormone and you think about it being hydrophilic, immediately think transmembrane protein receptor. If we see a hormone, and we're going to do this in a couple of minutes, when you see a hormone that's hydrophobic and therefore lipophilic, think cytoplasmic receptors. Divide your world into those 2 great categories. For the most part, we're going to be dealing with these hydrophilic hormones. The other second kind of hormone we can have is a modified amino acid. Of the 20 amino acids, only 2 can be modified into hormones. The first one we modify into hormone is tryptophan. and we modify it into serotonin. Another name for serotonin is 5-hydroxytryptamine. So the 5-hydroxytryptamine is the same thing as serotonin. This is an important neurotransmitter, important hormone in the brain. It's important in platelets and blood clotting. It does a whole number of things. We're going to leave it behind for the most part. The one I want to spend more time on as a modified amino acid is tyrosine. Now, I'll never memorize the structure, but I want to show you the structure of tyrosine because it's important to think about how it functions. And you'll remember when we talk about an amino acid, it has the amine, the carbon double bonded oxygen, OH. This is the carboxylic acid. Then, in this particular molecule, there's a carbon and a hydrogen and a hydrogen. And then down here is a ring structure. This is a six-membered ring. If this were the end of the structure, it would be phenylalanine. But in fact, we modified the phenylalanine down here to have a hydroxyl. So that's the structure of tyrosine. And the reason I put it up here is I want to point out that this hydroxyl makes the tyrosine polar. And because the tyrosine is polar, it's now hydrophilic, it's water soluble. If we take this hydroxyl off, we have phenylalanine, we have one of the hydrophobic amino acids. But as soon as we add the polar hydroxyl, this polar alcohol, to the ring structure, it becomes hydrophilic. It's tyrosine. Well, the tyrosine, this structure can be modified into the catecholamines. We'll modify try again. We'll modify this tyrosine into dopamine, epinephrine, and norepinephrine. I'm not going to go through the synthetic pathway here. If you take a neurochemistry course, you'll probably learn all of this, but I just want you to recognize that we can take tyrosine and we can convert them into any number of these catecholamines, dopamine, epinephrine, or norepinephrine. The distinguishing feature between epinephrine and norepinephrine is simply the addition of a methyl group. So these are all similar molecules. They're very similar, and they're derived from tyrosine. All of these, the catecholamines, have the characteristic of being hydrophilic. Because they're hydrophilic, they function through transmembrane protein receptors. We'll see on Monday that these are particularly the G protein coupled receptors. But tyrosine can also be modified in a different way. It can be modified into something known as thyroid hormone. In the thyroid gland, tyrosine is modified into thyroid hormone, and I'll just show you the structure, never ask you to repeat it, but it's based on the amine here, the carbon, hydrogen, the C double bonded O, OH. There's the carboxylic acid. Here's the CH2. Here's the ring structure. Oh. So we're going to put the ring structure in here, just as we saw for tyrosine. We're going to modify this just a little bit so that Where there was a hydroxyl here, this now goes to an oxygen. And now we hook it up to another tyrosine molecule. So now there is this next ring structure. This is the CH2. It then comes out here to the carbon, to the C double bonded O, OH, and the NH2. Now, all I'm asking you to look for here is the fact and recognize that this is one tyrosine molecule, this is another tyrosine molecule, and they're linked by this oxygen. Turns out in the process of making thyroid hormone, we also add iodine. The one I'm showing you here is something called T4 because there are 4 iodines. And another active form of this is T3 with 3 iodines. We're not going to get too into the weeds on that. But all I want to show you here is that by the time we add these big bulky iodine groups and we link the 2 tyrosines through this oxygen, this now becomes hydrophobic. So the thyroid hormone is a hydrophobic hormone. When you see hydrophobic hormone, immediately think cytoplasmic receptor. When we say here that this is a cytoplasmic receptor, all we're saying is that in contrast to these hydrophilic hormones which bind to transmembrane protein receptors, these cytoplasmic receptors are inside the cell. And they can bind to these hydrophobic receptors because the hydrophobic receptors are so lipophilic that they easily go into solution. They basically dissolve in the phospholipid bilayer and are easily shuttled then into the cell where they'll bind to the cytoplasmic receptor. Questions to there?
Maybe just clarify where the R groups are here.
Oh, okay. Where are the R groups? Here on tyrosine, here's the R group. That whole structure is an R group. This used to be the R group for tyrosine. Now we've taken the other tyrosine, turned it upside down, and used the hydroxyls on both of them and linked this oxygen to the two ring structures on the 2 tyrosines. So here, this would be an R group for this. Would be the R group for this tyrosine. This would be the R group for this tyrosine. We then use some enzymes to bring these together and form this oxygen bond between the 2 of them. And because the electrons are shared here, there's not much in the way of polarity. We've lost the polarity of the tyrosine by sharing the electrons between the 2 ring structures through that oxygen. Okay? All right. So over here, let me tell the story of the steroid hormones. The steroid hormones, again, are going to be hydrophobic hormones, and they're hydrophobic because they're based on cholesterol. Cholesterol is this ring structure. It's largely has all of its electrons shared. It has a whole bunch of side groups on it. We're not going to talk about the side groups, although if you take an endocrine course, you're going to learn about how to modify the side groups and change the cholesterol molecule into any number of different steroid hormones. But here in this panel, we're simply going to say that steroid hormones are derived from cholesterol. I'm going to draw the backbone of cholesterol for you here. Looks a little like this. And we number these rings A, B, C, and D. D is a 5-membered ring. Don't worry about the structure. But if you would look at all of the side groups on this, what you come up with is that for the most part, we see the electrons are equally shared. That is to say, this molecule is hydrophobic. This hydrophobic molecule now is not easily solubilized in water, but it can be modified into the variety of steroid hormones. It can be modified into the so-called sex hormones. I listed these in the introduction. I won't list them again. These will be testosterone. These will be estradiol and progesterone. All of those are sex hormones. They're secreted by either the testes or the ovaries, and they're all modified cholesterol. The other modified cholesterol is going to be vitamin D, also known as calciferol. The calciferol is important for calcium uptake, as we talked about yesterday. And the third type are the glucocorticoids. And under this heading of the glucocorticoids will be corticosterone, and cortisol, and a related one called aldosterone. We're going to run into aldosterone later when we talk about ion balance and aldosterone functions in the kidney. Corticosteroids are really important to us. We use them to respond to stress. So when we're stressed, our cortisol levels go way up. But we can use them medically as well, because the corticosteroids, both cortisol and corticosterone, the cortisols are anti-inflammatories. These are one of the best tools, anti-inflammatory tools we have, and we commonly use them when there's inflammation. Sometimes it's inflammation in joints, and we'll inject a joint with cortisol or corticosterone, and that's anti-inflammatory. One of the places you're going to see it commonly now is in the treatment of COVID, because a type of artificial, not a non-biological, but a synthetic form of cortisol is called dexamethasone. And dexamethasone is turning out to be one of the really good treatments for people who are deep into COVID and are starting to now have a problem of inflammation in their lungs and problem of overreaction of their immune system. And it seems to be that the corticosteroids, particularly in the form of dexamethasone, are going to be effective in treating those cases. The last piece here I want to talk about, the last couple of minutes, is I want to talk about the concept of affinity and specificity and KD. And this is an important idea in hormones, because the hormone binds to the receptor, and that's not a covalent interaction, that's a charge-charge interaction. And so the hormone binds to the receptor. We form a hormone receptor complex. The hormone receptor complex obeys all the rules of laws of mass action, so we can assemble and disassemble that hormone receptor complex. And I will spend a little bit of time talking about that in the next few minutes. So we can talk about the law of mass action informing the hormone receptor complex. And as I said in the introduction, we have a concentration of hormone plus a concentration of receptor that gives rise to a hormone receptor complex. But it's important to recognize that this hormone receptor complex can come undone into its component parts, so this reaction goes both ways. That's the idea of law of mass action. And it can go in both ways because fundamentally what we're looking at here, let's do it this way, so if this is the receptor, the hormone binds to the receptor to form the hormone receptor complex. But some really important ideas have to be met in forming this hormone receptor complex. And that is, the first one is affinity. What we mean by affinity is really nothing more than stickiness. How sticky is this receptor for the hormone? How tightly will they bind together? How effective is the charge-charge interaction of the three-dimensional fit of the hormone to the receptor? That's the idea of affinity. But in addition to affinity, it's not enough simply to have the hormone be sticky to the receptor. There has to be the concept of specificity. And what we mean by specificity is that there is a specific interaction between the hormone and its receptor. The specificity is in some ways related to the affinity, but this has to be a proper three-dimensional lock and key fit between the binding pocket on the receptor and the hormone. So there has to be this careful lock and key fit to form this specific binding between the hormone and the receptor. The last piece I want to talk about, I'm a little over here, but let me talk about, just in a moment, let me talk about KD. KD is defined as the concentration of hormone at which one half of the receptors are occupied. Let me start this here and then we can finish it on Monday. I'm going to draw out here a single cell. And then I'm going to put across this cell 4 receptor molecules. And down here in red, I'm going to put in red the hormone. Now let's say the concentration of hormone, let's just say for grins, it's 10 to the minus 9th molar. And let's say for this receptor, the KD is 10 to the minus 9th molar. So the KD for the receptor is 10 to the minus 9th molar, and we have 10 to the minus 9th molar hormone out here. How many of these receptors will be occupied? Simply by the definition. Half of them. Half of 4 is 2. Well, so that means there we are. But let's change the story just a little bit. Let's change the story and say that rather than the KD of the receptor being 10 to the minus 9th, let's say now it's 10 to the minus 6th. So now the KD is 10 to the minus 6th, but we're going to leave the hormone concentration at 1,000 times lower than the KD. So that means the hormone concentration out here is 10 to the minus 9th molar, but the KD is 10 to the minus 6th. How many of these receptors are likely to be occupied under those circumstances? Almost none, because in order to have half of these receptors occupied, we need a concentration of hormone of 10 to the minus 6th molar, but we only have 10 to the minus 9th. We can change this all the way around. We can say, well, let's now say the KD is 10 to the minus 9th. And let's take an extreme case and say the concentration of the hormone is 10 to the minus 3. So there are 1,000,000 times more hormone molecules out here than the KD for the receptor. So under these circumstances, since we have 1,000,000 times more hormone than we have the KD for the receptor, almost all of these receptors under those circumstances will be occupied. That's the concept of KD. And if you take a biochemistry course, a physical biochemistry course, you'll learn how to manipulate these numbers mathematically over and over and over again. All I want you to understand is the very concept of KD, the dissociation concept, the concept of where half of the receptors are being occupied, and concepts of specificity and affinity. With that, I'll let you go.